All right. My name is Noah Lyons. My research is focused on antibacterial resistance. Now, if you've ever taken a drug such as penicillin, amoxicillin, augmentin, all those things are what we classify as beta-lactam antibacterial drugs. I don't like saying antibiotic because you can't be against life. So we can be against bacteria though. So those drugs, what they do is they kill the bacteria. They have a cell wall surrounding them, and what it does is there's a sugar in there. And those drugs break up that sugar and make the bacteria so unstable that they end up just collapsing in on themselves and breaking apart. But bacteria are smart. They may not be living in the sense that we are, but they have defense mechanisms. So when we introduce these drugs, they have this enzyme, and we call it enzyme beta-lactamase. And that name just refers to the kind of compound that's in those drugs that it gets rid of. And so what they do is these enzymes, they come in and they take those drugs and they just make them inactive. They don't work anymore. And so what I was trying to do is saying, okay, we have these drugs, how can we make them better? So I synthesized these compounds, and what they have is a unique kind of structure that isn't the square beta-lactam structure that those lactamases normally hydrolyze. What they actually have is a 5-ring structure, and all that means is just that it makes it more stable. It makes it able to actually work better on the enzyme. So I designed the experiment, and I synthesized all but one thing from scratch. We used a commercially available compound, but the rest we made in the lab. So we did several different reactions of this, and we synthesized these compounds very well. They turned out very nice. Some of them were green, yellow, but the colors just indicated what kind of compounds we use. So I wanted to know, do these compounds actually cause inhibition? And inhibition is just a fancy word for saying the enzyme's not working. So I designed this enzyme study, and we took a known penicillin derivative called nitrocephin, and it's yellow when it's normal, and it turns red when that lactamase, that enzyme, cleaves that. So I ran several studies, and we found that 2 of our compounds were near 30 to 40% inhibition, which just means that about half that enzyme is broken up. And so what we discovered is that there are certain compounds that are more reactive than the other, and that we should be able to modify those in the future to get more progress toward this resistance. So if I were to sum everything up, I said the research we're doing is trying to make these beta-lactam antibacterials better. We are showing inhibition of that enzyme, and we're making sure that this enzyme is not working and we can use our drugs.